welcome to the deep dive.
Yeah.
You've handed us this incredible stack of sources, and our mission today is clear: to plunge headfirst into the hidden world of microbial ecosystems.
That's right.
These aren't just tiny organisms. They're vast, often invisible communities living everywhere, truly shaping our planet.
Exactly. We've got a rich collection of readings here covering microbes in soils, fresh waters, oceans, and some truly extreme spots like the deep subsurface and hydrothermal vents. Our goal is to pull out the most important insights, the surprising facts, the stuff that makes you realize just how fundamental this microscopic life is to everything.
And trust me, you will be amazed. As these sources reveal, we're talking about life with incredible diversity and adaptability. Microbes live in places no other life forms could possibly dream of surviving. From waters hotter than boiling, deep within solid rock,
Yeah, it's incredible.
to the crushing pressure of the deepest ocean trenches. Let's get into it.
Okay, let's start with some foundational ideas from the sources about how microbial life is organized in nature. Okay. When we talk about a group of microbes of the exact same species, all living in the same spot at the same time, that's a microbial population.
Simple enough. And when you have multiple different populations sharing a space, interacting, that's a community.
Right. And within those communities, you'll find guilds.
Guilds.
Think of a guild as a group of different populations that are all metabolically similar. They're essentially using the same resources, doing the same kind of job, in that ecosystem.
So different species, but the same profession, you could say.
Precisely. And the specific little corner of the habitat that a guild occupies, providing the resources and conditions they need to thrive, is called a niche.
Population, community, guild, niche, the basic units.
Got it. Now, something the sources really drive home is the concept of the microenvironment.
Oh, this is absolutely crucial because microbes are so incredibly small.
Right.
The conditions they actually experience are just in their immediate local surroundings. And that tiny space can be dramatically different from the overall habitat. Like the example from the source about oxygen levels inside a single soil particle.
Exactly that one. On the outside, it's totally oxygenated, like the air.
Sure.
But just a few millimeters inside, microbes on the edge gobble up all the oxygen, and it can become completely anoxic or oxygen-free.
Yeah. So a few millimeters.
Yeah, and the analogy they use is brilliant. For a tiny 3-millimeter rod-shaped bacterium, a distance of just 3 millimeters is like 2 kilometers to a human.
Whoa.
It really helps you picture that scale and how varied conditions can be within what looks uniform to us.
Okay, so even a tiny soil clump isn't uniform at the microbial level. You can have completely different living conditions right next to each other.
Exactly. This microscale variation, along with nutrient availability and overall conditions, heavily shapes the species' richness.
That's the total number of different species.
Right. In species abundance, how many individuals of each species are there?
Okay.
Interestingly, extreme environments often have low richness. Only a few tough species can hack it.
Makes sense.
But those few species can be incredibly abundant.
Like the acid mine runoff bacteria we've heard about, dominating a superacidic, iron-rich niche where nothing else can survive.
They have the place pretty much to themselves, yeah. Now, the sources emphasize that microbial life in nature is often less like a predictable farm and more like a feast or famine existence.
Feast or famine? How so?
Nutrients might show up in pulses, then followed by long periods of scarcity.
Not the stable, perfect world of a lab culture.
Definitely not. And this is why microbial growth rates in nature are typically way, way slower than what we see in the lab.
Really?
Yeah, the source gives striking examples. E. coli in your gut has a generation time of around 12 hours.
Okay.
But in ideal lab conditions, it can double in 20 minutes.
Huge difference.
And soil bacteria in nature are estimated to grow at less than 1% of their maximal lab rate.
Less than 1%. That's astonishing. Why the massive difference?
Well, it's a combination of factors. Conditions are rarely optimal everywhere. Nutrient distribution is patchy. Remember that microenvironment?
Right.
And importantly, they're not growing alone. They're in complex communities, constantly competing for those patchy resources.
Competition.
Which naturally slows things down compared to a pure, pampered culture.
Competition and patchy food supply make perfect sense. Okay, before we dive into specific habitats, the sources touch on biogeochemical cycles.
Right. These are the transformations of essential elements like sulfur, nitrogen, carbon, phosphorus, iron.
Big ones.
Yeah. Microbes are often the key players, the catalysts driving these cycles.
And they're also called nutrient cycles, aren't they? Because they recycle things.
Yes, exactly, because one of the most critical roles microbes play is regenerating forms of these elements that are needed by other organisms, especially plants.
Hmm.
They essentially recycle the planet's building blocks. These cycles frequently involve coupled oxidation and reduction reactions, constantly moving elements between different chemical states.
Making sure the essential stuff is available for everyone else.
Hmm.
Okay, let's start our tour with terrestrial environments, specifically soils.
Okay, soil. It's far more than just dirt. The sources define it as the loose outer layer, distinct from bedrock,
Right.
that forms over vast periods through a complex interplay of geological materials, topography, climate, and crucially, living organisms.
It's a whole active system, and in vegetated soils, you have this mix. About 40% inorganic minerals, maybe 5% organic matter, 50% air and water,
Mm.
and that crucial 5% that's teeming with microorganisms and macroorganisms.
And if you look at a soil profile, the layers called horizons, O, A, B, C, the highest numbers and activity of microbes are concentrated in those organic-rich upper layers, the O and A horizons.
Where all the good stuff is.
Exactly. You can find up to 10 billion cells per gram in the O horizon.
10 billion in a pinch of soil.
Think about that. Billions of living things.
So what are the key processes creating and sustaining all this life in the soil?
Well, it's a beautiful dance between physical, chemical, and biological processes. You have pioneering organisms like algae and lichens colonizing bare rock, starting to create organic matter.
First step?
Then chemorganotrophs break that down. Chemical reactions like CO2 from respiration forming carbonic acid,
Oh.
or organic acids secreted by microbes and roots help dissolve rock. Physical processes like freezing and thawing fragment rock.
Makes sense.
Plant roots are major players too. They break up rock and support massive microbial populations right around them in the rhizosphere.
The rhizosphere. That's the zone right around the roots.
Yeah, thanks to the nutrients the plants secrete there. And of course, dead plants and animals constantly add more organic matter back in.
A perpetual cycle driven from the microscopic up. How does water availability affect things in soil?
Water is critical. It exists in thin films absorbed onto particle surfaces or fills the spaces between particles. This is where the microenvironments really come alive again. Well-drained soil had lots of air pockets and is oxic.
Plenty of oxygen.
But if it gets waterlogged, those air spaces fill, oxygen diffusion slows to a crawl, and it quickly becomes anoxic,
Right.
favoring completely different microbial groups and metabolisms.
Another example of tiny spaces. Massive differences. The sources also mention microbial life in very dry places.
Yes, habitats like biological soil crusts, or BSCs, which are found on the surface in dry lands, or microbes living under rocks hypolithic, on rocks epilithic, or even inside rocks endolithic.
Inside rocks.
Yeah. BSCs are particularly important, often dominated by filamentous cyanobacteria like Microcoleus. These cyanobacteria excrete sticky sheath material.
Like a glue.
Sort of, yeah. It binds loose soil particles together, forming a crust. They also perform photosynthesis, bringing in organic carbon, supporting the entire community beneath them.
So even in deserts, microbes are literally holding the landscape together and providing the food base. That's pretty incredible. Let's talk more about the sheer diversity in soils.
Studying soil microbes directly is challenging, but techniques like fluorescence microscopy, gene probes like FES, and electron microscopy give us glimpses.
Okay.
Looking at bacterial composition, the dominant groups, or phyla, are typically proteobacteria, acetobacteria, and bacteroidetes.
Why are those three groups so prevalent? What makes them successful there?
Their success is tied to their incredible metabolic flexibility. They can utilize a huge variety of organic compounds found in soil.
Like what?
Oh, everything from simple plant sugars to complex polymers. Groups like acetobacteria are good at handling fluctuating oxygen levels.
Right, the oxygen-oxic thing.
Yeah, they can use things like iron, nitrate, or sulfate as electron acceptors when oxygen is scarce. Bacteroidates are particularly good at breaking down complex carbohydrates.
Gotcha. And groups like actinobacteria and firmicutes are less diverse in soil comparatively. What about archaea?
Archaea diversity in soil is generally lower, mostly Euryarchaeota and Thaumarchaeota.
Okay.
But here's where a surprising insight from the sources comes in. Despite lower diversity, Thaumarchaeota can be incredibly important.
How so?
They include ammonia-oxidizing archaea, and they're often thought to be responsible for the majority of nitrification, a critical step in the nitrogen cycle.
Right, making nitrogen available to plants.
Exactly, in most soils, even outnumbering the ammonia-oxidizing bacteria.
So low number of types doesn't mean low impact. There may be few in species, but mighty in numbers and critical function for plant growth.
Precisely. And the sources highlight how pollution, like hydrocarbon spills, can really mess with this balance.
Oh.
It changes the proportions of these major groups and, worryingly, suppresses those key ammonia-oxidizing thaumarchaeota.
That's a significant point mentioned in the sources, the potential threat to global food supplies if climate change or pollution alters soil microbial diversity, given how critical they are for nutrient cycling.
It really underscores how much we rely on these unseen processes. Now let's shift our focus from the surface soil downwards into the terrestrial deep subsurface.
Deep subsurface, everything below that sea horizon bedrock transition.
Yeah.
I can only imagine how different it is down there. Dark, probably low on everything.
Very different. Much lower nutrient levels, significantly lower diversity, and lower cell abundance compared to surface soils.
So how do they survive?
Microbes survive by scavenging what little organic carbon makes it down, mostly from dead cells. But a crucial energy source is chemolithotrophy.
Using chemical energy from inorganic compounds, right? Not organic stuff.
Yes, exactly. They tap into inorganic electron donors found in the rock or fluids, things like hydrogen gas.
Hydrogen gas, where does that come from deep underground?
Well, it can be generated by the radiolysis of water, basically. Water molecules being split by radiation from radioactive elements in the rock, or from geochemical processes where minerals like iron silicates are oxidized.
The sources give that incredible example from South Africa, don't they?
Oh yeah, the bacterium Desphosciutus audaxviator found 3 kilometers deep in a gold mine.
3 kilometers.
It's never been cultured, but its genome revealed it's perfectly adapted for extreme isolation. It uses hydrogen, reduces sulfate for energy, can fix CO2 for its carbon and nitrogen from N2 gas.
Wow.
It's built for a lean diet, living completely cut off from the surface world, powered by energy locked in the rock itself.
An entire self-contained ecosystem powered by geochemistry and microbes, kilometers underground. Mind-boggling.
And the archaea down there are equally fascinating groups like Bathyarchaeota, the Asgard group, the Deepan superphylum. Many are uncultured, some are extremely small, potentially symbionts. They seem to have completely novel metabolisms involved in things like carbon degradation or methane oxidation.
The Asgard group. Isn't that the one linked to the origin of eukaryotes, like us? I think I read something about that.
Yes, that's the one. Their genomes have been found to encode proteins that are very similar to those found in eukaryotic cells, like actin.
Seriously?
Yeah. This provides strong evidence that the eukaryotic domain, everything from fungi and plants to animals, likely emerged from within the archaea, specifically somewhere related to the Asgard superphylum.
That's a fundamental connection to our own deep past, buried deep underground. Amazing. And these microbes grow incredibly slowly. Is that right?
Incredibly. We're talking generation times that can range from days to potentially centuries for a single cell division in some cases.
That's reasonable.
Yeah. Life at the absolute edge of existence.
Okay, let's leave the solid earth behind and dive into aquatic environments, starting with freshwaters, lakes, streams, rivers.
Okay, freshwaters. They're incredibly dynamic. They range from crystal clear mountain lakes to turbid, nutrient-rich rivers heavily impacted by runoff. Resources and conditions can vary dramatically.
The primary producers must be key here, generating the energy base just like on land.
Absolutely. These are oxygenic phototrophs organisms that use sunlight and produce oxygen like algae and cyanobacteria.
Right.
They can be planktonic, floating freely, or benthic, attached to surfaces. They're essential because they create the organic matter and the oxygen that fuel the rest of the food web.
And the balance between that oxygen production and the oxygen consumed by other microbes determines the water's oxygen levels, sort of like in the soil particle.
Exactly the same principle. If there's lots of primary production, you get oxygen. But if there's also a lot of organic matter coming in, say, from pollution or dead algae, microbes consuming that organic matter can deplete oxygen, especially in deeper waters, leading to anoxic conditions that favor anaerobic metabolism.
And seasons matter too.
Oh, yes. Things like seasonal stratification in temperate lakes where water layers form and don't mix well, or sudden events like storms drastically affect how nutrients and oxygen are distributed.
This is where biochemical oxygen demand, or BOD, comes in, right?
Right.
I remember seeing that term.
Yes, BOD is essentially a measure of how much oxygen microbes will consume in a water sample. It indicates the amount of oxidizable organic matter present.
So high BOD means Lots of food for microbes.
Exactly. And the sources show that where you have high BOD, like from sewage input, the dissolved oxygen levels plummet. Microbes are feasting on the organic matter and using up the oxygen rapidly.
More microbial food means less oxygen for everything else that needs it, like fish.
Got it.
What does the microbial diversity look like in freshwaters?
Surveys of freshwater lakes show major bacterial groups like proteobacteria, actinobacteria, bacteroidetes, cyanobacteria, and verrucomicrobia. Archaea are also present: Euryarchaeota, Crenarchaeota, Thalmarchaeota, but usually in lower numbers than bacteria.
So some overlap with soil, like proteobacteria and bacteroidetes. Any key differences?
Yes, beta proteobacteria, a subgroup of proteobacteria, are often more prominent in freshwaters compared to marine environments. The diversity here is linked to the dynamic nutrient inputs and the metabolic versatility needed to handle changing conditions.
Like what sort of versatility?
Well, you have bacteroidetes breaking down complex stuff, actinobacteria working on nucleic acids and proteins, potentially even some unexpected groups like phototrophic actinobacteria or anoxygenic phototrophic proteobacteria.
Interesting.
And of course, cyanobacteria can form those visible blooms when nutrients are high.
That sounds like freshwater microbes are constantly adapting to a variable environment. Okay, let's head into the vast blue marine environment, starting with the open ocean, the pelagic zone.
Right, the open ocean, particularly the vast central gyres, it's fundamentally different from freshwater. It's generally much lower in nutrients, especially nitrogen, phosphorus, and iron, although oxygen is usually plentiful near the surface. Temperatures are also more stable and cooler than many freshwaters.
And cell numbers are typically lower per volume, right? Fewer microbes packed in.
That's correct. Around 106 cells per milliliter in the open ocean compared to roughly 107 in many freshwaters.
Okay.
But despite the lower density, marine microbes are absolutely critical globally. They consume the largest pool of organic carbon on Earth, the dissolved organic carbon in seawater.
Oh.
And they regenerate nutrients, basically taking dead stuff and recycling it. This secondary production is key to moving energy and nutrients up the food web.
They're the unseen recyclers of the ocean. Essential.
Exactly. Long-term studies, like the one mentioned near Bermuda, track how these communities change seasonally, linked to things like spring phytoplankton blooms or summer stratification of the water column.
And many open ocean microbes are tiny, built for low nutrients. Is that an adaptation?
Yes, they are often oligotrophs, adapted to thrive in nutrient-poor conditions. Being small helps less energy needed for basic maintenance.
Ah, efficiency.
Yeah, and a higher surface area to volume ratio, which is great for scavenging scarce nutrients. Their genomes often reflect this, being streamlined or very compact, encoding high-affinity transport systems to grab onto those precious few nutrient molecules.
Efficiency is everything when food is scarce. Makes sense. Who are the big players among the phototrophs out there? The ones making food from sunlight.
A truly massive player in the open ocean is the cyanobacterium Prochlorococcus.
Prochlorococcus, okay.
It's estimated to contribute up to half of the total primary production in tropical and subtropical waters globally.
Half from this one tiny microbe.
It's incredible, and it's unique, incredibly tiny, and olive green because it lacks the usual blue phycobilin pigments of other cyanobacteria, but still has chlorophylls A and B.
Olive green, interesting. And the sources mention different ecotypes, like variations.
Yes, there are different genetic variants or ecotypes of Prochlorococcus specifically adapted to different light intensities and nutrient levels.
So they specialize.
Exactly. You find highlight adapted types near the surface and low light types deeper down, sometimes even extending into the upper part of oxygen minimum zones down to 200 meters.
Single-cell genomics must have revealed a lot about them.
A tremendous amount. It showed incredible fine-scale genetic diversity even within what look like similar ecotypes. They have a core set of essential genes, but also blocks of flexible genes often located in genomic islands. These flexible genes give them versatility, allowing them to adapt quickly to seasonal changes in light, nutrients, or threats from predators.
So they have this stable core, but can swap out modules in their genetic toolkit to handle changing conditions. That is so clever.
It really is.
Yeah.
Other oxygenic phototrophs include the filamentous cyanobacterium trichodesmium, which is important because it can fix atmospheric nitrogen, forming visible colonies or puffs. And surprisingly tiny eukaryotic algae like Ostriococcus, one of the smallest eukaryotes known.
How small?
About the same size as an E. coli bacterium at just 0.7 micrometers in diameter.
A eukaryotic cell that small is wild. Usually they're much bigger.
Yes. And then there's a fascinating group called the aerobic and oxygenic phototrophs, like Rosiobacter.
Aerobic and oxygenic. That sounds contradictory.
It does. These are proteobacteria that contain bacterial chlorophyll, a pigment usually associated with photosynthesis.
Right.
But they only perform photophosphorylation using light energy to make ATP, the cell's energy currency, under oxic conditions.
So they use light, but only when oxygen is around. That's not what I expected for photosynthesis. They don't make oxygen.
No, they don't make oxygen, and crucially, they are photoheterotrophs. They still need to consume organic carbon for growth. They don't fix CO2 like classical phototrophs, but they use light energy through photophosphorylation to generate extra ATP, supplementing their chemorganotrophic or organic carbon-consuming metabolism. It's an energy booster when food is scarce.
Supplementing their diet with sunlight that's brilliant for a low-nutrient environment. Looking at the overall microbial picture, how are bacteria and archaea distributed with depth? Is it even?
Bacteria generally dominate in the upper 1,000 meters or so, but below that, bacteria and archaea are often found in near equal abundance.
Interesting shift. And the sources mention one bacterium that might be the most abundant cell on Earth.
Yes, Pelagibacter, part of the Serralivin group within the alpha proteobacteria. It's estimated there are a staggering 2.4 by 1,028 cells of Pelagibacter globally.
24,000 trillion cells. That number is almost impossible to wrap your head around.
It really is, and it's a classic example of an oligotroph, with those high-affinity transporters and a streamlined genome perfectly tuned for the open ocean's low-nutrient environment.
And many marine microbes, including Pelagibacter, have proteorhodopsins. What are those?
This is another cool adaptation. Proteorhodopsins are light-driven proton pumps found in the membranes of many marine microbes.
Proton pumps, like they move protons.
Yeah, they capture light energy and use it to pump protons across the membrane, generating a proton gradient that can be used to make ATP.
So like the aerobic and oxygenic guys, using light for energy, but not full photosynthesis.
Exactly. Again, this isn't photosynthesis in the classical sense of fixing CO2. It's using light just for energy conservation, supplementing their ATP production when organic carbon is low. Different versions absorb different light wavelengths, adapted for different depths. The sources also mention heliodopsins, which might act as light sensors.
So light is a usable energy source even for microbes that aren't typically thought of as photosynthetic. Fascinating. What about the major bacterial groups overall in the ocean?
Alpha and gamma proteobacteria are generally the dominant groups. They play diverse roles, for example, metabolizing DMSP, dimethylsulfoniopropanid. It's a sulfur compound produced by phytoplankton, which releases DMS when broken down. DMS can enter the atmosphere and impact climate by contributing to cloud formation.
Wow, microbes affecting clouds.
Yeah. The sources note regional differences. Prochlorococcus and other cyanobacteria dominate the open ocean, but are less common in coastal areas due to competition. There's also the candidate phylum Microbia, which is very widespread and seems metabolically flexible, including capabilities for anaerobic respiration.
Yeah, marine archaea. We talked about them in soil.
You find Euryarchaeota, Crenarchaeota, and Thalmarchaeota here too. As in soil, the ammonia-oxidizing Thalmarchaeota, like Nitrus apumilis, are really abundant in deep water below the photic zone.
How abundant?
Making up 20–40% of prokaryotes there, where they play a key role linking carbon and nitrogen cycles. Some surface Euryarchaeota even contain proteorhodopsins, likely functioning as photoheterotrophs like the bacteria we discussed.
Okay, let's talk about oxygen minimum zones, or OMZs. What exactly are they?
These are distinct layers in the ocean, typically found between 100 and 1,000 meters depth, characterized by very low oxygen.
Low oxygen. Why there?
They form in areas with high surface primary production. When that organic matter sinks, microbial respiration consumes oxygen faster than it can be replaced from the surface or deeper waters.
And there are natural OMZs and ones caused by human activity, like dead zones.
Yes, natural OMZs are part of ocean circulation. But human-caused marine dead zones, like the large one in the Gulf of Mexico, are driven by excessive nutrient runoff, primarily from agriculture.
Right, the fertilizer runoff issue.
Exactly. This fuels massive algal blooms. When they die and sink, their decomposition creates severe oxygen depletion.
What's the ecological importance of OMZs, besides being bad for fish, I guess?
Well, there's significant sinks for fixed nitrogen in the ocean through anaerobic processes like denitrification and anammox, but they can also be sources of N2O, nitrous oxide.
That's a potent greenhouse gas, isn't it?
It is.
And the sources link the expansion of OMZs to climate change. How does that work?
Exactly. Warmer surface waters become more stratified, meaning they mix less with the deeper layers.
Less mixing.
Yeah. This reduces the supply of oxygen to deeper water, causing OMZs to expand naturally and likely worsening the human-caused ones. This expansion impacts aerobic life, including fish, and could threaten fisheries.
The sources also briefly mention how microbial communities responded to the Deepwater Horizon oil spill.
They did. Hydrocarbon-degrading microbes became incredibly abundant, basically blooming in response to the oil, helping to break it down. A rapid microbial response to a disaster.
It's amazing how these microscopic dynamics are connected to global processes, climate, and even environmental disasters. What about the viruses in the marine environment? They get mentioned too.
Viruses are vastly important players that we often overlook. They outnumber cells by orders of magnitude.
Really? Outnumber them?
Oh yeah, hugely. And they are major forces in marine food webs. By killing their host to lysis, they release huge amounts of dissolved organic carbon.
Food for others.
Yes, exactly. Which then becomes available for other microbes to consume. They're also key agents of genetic transfer between cells through transduction, moving genes around.
And they carry these things called auxiliary metabolic genes, or AMGs. That seems interesting.
This is a really fascinating point from the sources. Phages, the viruses that infect bacteria, often carry AMGs.
What are they?
These are genes that are not needed for the virus's basic replication cycle, but they can alter the host's metabolism in ways that ultimately benefit the virus.
Benefit the virus. How?
For example, phages that infect Synechococcus and Prochlorococcus cyanobacteria often carry photosynthesis genes, specifically for photosystem II.
Right. The virus has photosynthesis genes. Why would a virus need those?
The theory is that by inserting these genes, the virus helps the infected host cell keep photosynthesizing and producing energy even while the virus is taking over its machinery.
So the virus keeps the host's lights on.
Exactly. This continued energy production helps the host build more viral particles more efficiently.
So the virus helps the host do photosynthesis better, so the host can make more virus. That is wild and slightly sinister.
Chuckle slightly. It is. Or, in low-phosphate waters, phages might carry genes for phosphate acquisition to ensure the host has enough phosphate to build all that new viral DNA.
Wow.
AMGs have even been found in viruses infecting Thaumarchiota archaea, including a gene for ammonia monoxygenase involved in their key ammonia oxidation metabolism.
So viruses are meddling with core biogeochemical cycles too.
Potentially, yes. Viral metagenomics is showing us a huge, mostly uncultured viral diversity out there with all sorts of potential AMGs.
This invisible viral world is constantly manipulating the microbial world it lives within.
Okay, let's take the final plunge into the deep sea, below about 1,000 meters.
Okay, the deep sea. This is a truly extreme environment. Bone-chillingly cold, maybe 2-3 degrees C.
Freezing.
Under immense pressure, extremely low nutrients, and completely dark. Life here is fundamentally chemotrophic, relying on chemical energy sources, not sunlight.
And they have to somehow deal with crushing pressure. How do they manage that? They do. Organisms here are often pisophiles, meaning they grow best under elevated pressure, greater than 1 atmosphere.
Okay.
Or even extreme pisophiles, which actually require hundreds of atmospheres of pressure to grow.
Require it. They can't grow without it.
Some can't, or grow very poorly. Examples include Photobacterium profundum, a pisophile from shallower deep sea, and Colwellia murnamaniae, a hyperpisophile isolated from the very bottom of the Mariana Trench.
Wow.
Thriving at 120 mpdi, that's over 17,000 psi of pressure.
How do their cells handle that? Do they have special pressure armor or something?
No, not armor, but molecular adaptations.
Huh.
The sources mention things like altering the composition of their cell membranes
Oh.
to have more unsaturated fatty acids, which helps keep the membrane fluid under high pressure. They also have pressure-sensitive proteins like the porin, which can alter gene expression in response to pressure.
So their genes turn on or off depending on the pressure.
Seems like it. Pressure can affect how proteins fold and interact, so their cellular machinery is specifically adapted. It's active adaptation, not just passive tolerance.
And like other extreme environments, there's still huge uncultured diversity down there. Lots we don't know.
Absolutely. Groups like Microbia, Gemmatimendidets, and those abundant ammonia-oxidizing archaea are found down here, many still only known from their genes.
Okay, moving from the deep water column to the deep sea sediments below the seafloor, that sounds even more remote.
It is arguably one of the largest and least explored biospheres on Earth. Studying it is incredibly difficult and expensive, requiring drilling cores deep into the seabed.
I bet. What's life like down there?
Cell numbers drop dramatically with depth in the sediment from maybe 109 cells per gram near the surface down to less than 103 cells per gram, hundreds of meters down.
Why the big drop?
Because the supply of organic carbon decreases and its quality gets poorer the deeper you go. Less food filtering down from above.
Fewer cells because there's less food. Are there any exceptions, places with more life in the sediments?
Yes, there are nutrient oases within the sediments, like methane seeps or coal beds, where chemical energy sources are more available.
What kind of energy sustains microbes deep within these sediments, away from those oases?
Well, they scavenge what little low-quality organic matter is there, use organic carbon from dead cells,
Yeah.
and potentially rely on abiotic sources. These could come from subsurface heating or geochemical reactions, releasing things like methane, hydrocarbons, hydrogen gas, acetate, or CO2.
So geochemistry again, like in the deep rock.
Potentially, yes. Their metabolisms are inferred to be mainly anaerobic, like sulfate reduction, fermentation, and possibly some totally unknown pathways. Growth rates here are probably also incredibly slow, maybe even slower than the deep subsurface rock, potentially centuries per division.
Just incredibly slow life. And the diversity in marine sediments, what groups dominate?
It's dominated by proteobacteria, particularly gamma, delta, and epsilon subgroups,
Really?
especially sulfate reducers like Desulfobacterales. Which makes sense because sulfate is abundant in the seawater that saturates the sediment. There's also a high proportion of novel archaea here. You find minimal cyanobacteria because it's dark and anoxic. Any are likely just dormant cells that settled from the surface.
Okay, finally, let's talk about the ultimate deep-sea energy hotspots.
Ah.
Hydrothermal vents. The smokers.
The vents, yes. These deep-sea hot springs are entirely powered by chemical energy, a classic example of chemolithotrophy driving an entire ecosystem.
Xenolithotrophy, using inorganic chemicals for energy.
Exactly. Microbes here use inorganic electron donors that pour out of the vents. Sulfide, ammonia, hydrogen, iron, manganese, methane, carbon monoxide. Lots of options.
Wow.
Many of these microbes are also autotrophic, meaning they fix CO2 from the surrounding water as their carbon source, building organic matter from scratch using chemical energy.
So life completely independent of sunlight, fueled by Earth's chemistry, building its own food base right there. And they live right in those intense temperature and chemical gradients around the vents. Must be harsh.
Extremely harsh. You find incredible life thriving in gradients from near-freezing deep sea water right up to superheated vent fluids, sometimes over 100 degrees C or even higher, right in the chimney walls.
Wow.
Domophilic and hyperthermophilic bacteria and archaea colonize the walls of smoker chimneys like Metanoporus, Pyrolobus, and Pyrodictium. Different types of vents like the hot, sulfide-rich black smokers versus the cooler, more alkaline carbonate vents like Lost City support different microbial communities.
Makes sense.
Often with archaea being more dominant in the hottest parts.
And the overall microbial diversity near vents, is it high or low?
You find high bacterial diversity actually dominated by proteobacteria, particularly epsilon proteobacteria. Archaea diversity is generally lower near the plume itself, but they can be very abundant in the extremely hot chimney structures. You find methanogens, green archaeota, euryarchaeota thriving in those temperatures.
It really is astonishing, the sheer range of environments microbes inhabit and the metabolic tricks they employ. From Antarctic rocky deserts and deep subsurface rock to freshwater lakes, the vast open oceans, the crushing depths, the sediments below the seafloor, and the chemical cauldrons of hydrothermal vents.
It's mind-blowing.
It is. These sources paint a picture of a planet utterly dominated by microbes. They are fundamental drivers of biogeochemical cycles, regenerating the nutrients essential for literally all other life.
Yeah.
They capture and transform energy in ways macroorganisms can't, influence climate, and are critical to our own health and ecosystems through things like biofilms and nutrient cycling.
And despite everything we've covered today, these readings also make it abundantly clear how much remains unknown. The vast majority of microbial diversity and activity, especially in those extreme and remote environments, is still waiting to be discovered and understood.
Absolutely. Considering their sheer numbers, billions in just a gram of soil, trillions upon trillions in the ocean, and their critical, often unseen roles in every single corner of the planet.
Providing the basic services that support all macroscale life, including our own.
It really makes you wonder, are we truly appreciating the microbial world as the fundamental engine of Earth's ecosystems? The foundation for everything else.
That's a powerful thought to sit with, really puts things in perspective.
Indeed. That's been our deep dive for today. Thanks for joining us.